All right, let me blow this up so you can see what I'm talking about here. So this is all about operons, and so that learning targets for this are: I can explain the concept of an operon as a method of bacterial or prokaryotic gene regulation. Okay, so what happens in eukaryotes, each gene has its own promoter. Okay, so gene number 2 would have its own, gene number 3. But in prokaryotes, what they do is they have these genes that have proteins that work together. So you'll never need protein number 2 without protein number 1 or number 3. And so you want to, it would make sense to make them all at the same time or not make them all at the same time. And so what bacteria do is they clump those genes under the same promoter. Promoter, remember, is where the RNA polymerase binds. For transcription, okay, and that's going to be the start of gene expression. Within that promoter, which is just a segment of DNA, I'm showing it like a block, but this is like A's, T's, C's, and G's, right? Just a bunch of A's, T's, C's, and G's, all of this. But within that, there is an operator, and that operator region is where a repressor can bind, okay? And a repressor is something that turns this off, and the reason it turns it off is because it prevents RNA polymerase from binding. So let's just look at this operon in the normal state. Okay, so I got little friends here. Here's RNA polymerase. It's going to bind to the promoter. Okay, and then it's just going to down. And as it does, it's going to transcribe this into a piece of RNA. Okay, and so that's the goal of gene expression, or if we want gene expression to occur, that has to happen. Okay, RNA has to bind, and it has to read the DNA and make the mRNA. Okay, so that's when the operon is on. Okay, when it's turned on, RNA polymerase can bind and it can do its job. When it's turned off, what happens is we bind a repressor to the operator, and now RNA polymerase can't bind, and even if it could, it can't get past this. This is like a roadblock. It's like, nope, can't go any further. And so that's how we turn these genes off, is by blocking RNA polymerase, not letting it do what it wants to do. Okay, so that is how bacteria control the expression of these genes within this operon. Okay, is they either have a repressor so RNA polymerase can't bind, or the repressor comes off and RNA polymerase can bind. So this is on, this is turned off. Okay, I can explain the functions of the components of an inducible operon. Okay, so with an inducible operons are able to be induced. Okay, so if you break the word down, they're able to be induced. So they are normally, in their natural state, off, because I'm going to be able to turn them on. So if I can turn them on, that means they must be off, okay? So they're normally off, which means the repressor is bound. Something comes in, we call that something an inducer. It binds to the repressor. Whoopsies. Oh, let me put this up here so you'll know what we're talking about. This is an inducible operon, okay? I made that too big, I didn't realize it. Okay, it binds to the repressor. That makes the repressor come off, okay, takes it off of there. And so the inducer removes the repressor. And now RNA polymerase can bind and do gene expression. Okay, so it's normally off, but we can add that inducer, which pulls off the repressor, and now it turns the gene on, or the operon on. Okay, so that's kind of how an inducible operon works. Okay, so I can explain the functions of the components. So the components are the promoter, which is where the RNA polymerase binds. Okay, the operator, which is where the repressor is bound, and the inducer, which binds to the repressor. So again, the repressor is here in your normal inducible operon. The inducer binds to it, pulls it off, okay, and now the gene is turned on. Okay, so those are the three main functions or parts, are the promoter, the operator, and the repressor, or I guess the inducer. All right, I can explain the functions of the components of a repressible operon. All right, a repressible operon, let's put that up there. Repressible means able to be repressed or able to be turned off. So these are going to be normally on, okay, in this case. And that means they're normally, you're transcribing and making whatever it is. But in this case, when we want to turn it off, we need a co-repressor. Whoops, activates repressor. Think about what co means, like you work with your co-workers, means together. So the co-repressor is going to come in. It's going to bind to the repressor, which is over here just hanging out. It's going to activate that repressor to bind to the operator. Okay, so it activates the repressor, so it turns the operon off. Okay, so normal state there, it's on, RNA polymerase can bind and do its thing. When we want to turn it off, the co-repressor binds to the repressor. It attaches to the operator, and now the RNA polymerase can't bind, and so that turns the operon off. So the three main parts, promoter, operator, and co-repressor. All right, I can give an example of a repressible operon. An example of a repressible operon is the tryptophan operon, or the TRIP operon. Tryptophan is a molecule, it's an amino acid that organisms need. It's one of the 20 amino acids. And so bacteria make tryptophan, so they produce enzymes that can form or make tryptophan in the normal situation. So this is what they're doing. Making tryptophan, making tryptophan, making tryptophan. Okay? They're making the enzymes that make tryptophan, but anyway. When I give the bacteria tryptophan, or when the environment gives the bacteria tryptophan for free, they don't want to spend energy making it. So that tryptophan will bind to the repressor, turning it on, and it will block this. So now I'm not making tryptophan because I'm getting it for free. It makes sense that tryptophan would be the co-repressor, because that way the only thing that would stop this is the presence of tryptophan, which again, if I'm getting tryptophan for free, I don't want to make it. Okay, so a tryptophan operon is one that produces enzymes that make tryptophan, which is necessary for life. So that's why they want to normally keep it on. But then when they're getting it for free, they want to be able to turn it off, and that's what they do, using tryptophan as the co-repressor to activate the repressor. Okay? Now let's look at an example of an inducible operon. An inducible operon is normally off. The lac operon is an example. Lac stands for lactose. So bacteria can digest lactose, which is a sugar, and use it for energy. But they don't want to make the enzymes to digest lactose unless they have lactose. So this is their normal state. Okay, they have not been fed, no lactose. They get fed lactose. Lactose comes in, it binds to the repressor, so it acts as the inducer, binding to the repressor, pulls it off, and now the bacteria can make the enzymes that are necessary to digest lactose. Okay, so it's normally off because we don't want to make those digestive enzymes if we don't have any food, but we want to be able to turn it on when we eat. And I say we, I'm talking about bacteria. We don't have operons, but put your mind in the brain of bacteria. Not that they have brains either, but you know what I mean. So that would be an inducible operon, and that's usually going to be with digestive type things, catabolic enzymes. All right, the last thing is compare and contrast inducible and repressible operons. So let's compare first. Both operons deal with a repressor, right? Whether it's on or off, they have a repressor. Both operons have a promoter, an operator, and functional genes, or just what we call genes. So those are ways that they're similar. Ways that they are different: repressible are normally on. Repressible use a co-repressor to turn the operon off. Repressible generally are anabolic in nature, meaning they're building things, like building that tryptophan. Okay. Then if we look at the inducible, they are normally off. Okay, so they are normally in this state. We have an inducer that comes in, so it has an inducer, whereas the repressible had the co-repressor. And they are generally catabolic, meaning to break down in nature. So those are at least 3 similarities and 3 differences. I hope that helps, but if you're still unsure about operons and how they work and what they are and the difference between these two, let me know and we'll go over it again. Thanks, guys.